Hello everyone. Today we are going to see our last module in biodiversity, that is the tools and techniques in taxonomy or tools and techniques in classification science. There are different characters in taxonomy, like morphological characters are there, cytological characters are there, ethological characters are there, ecological characters, and then biochemical characters. So based on these characters, we generally classify the organism, and we can understand the contents and their characteristics of the organism. First of all, we need to know what is meant by molecular taxonomy. The classification of organism on the basis of distribution and composition of chemical substances in them, and the molecular taxonomy. So what comes under the molecules is the DNA, RNA, and proteins. So DNA is the blueprints of life, like each and every information is conserved or which is stored in the DNA material, and it transcribe into the RNA and then ultimately translate into protein. So these process is called it as central dogma of life. So whatever proteins are present in the organism, that ultimately has to come from the DNA material. So molecular techniques in the field of biology have helped to establish the genetic relationship between the members of different taxonomic characteristics. So, molecular phylogenetics. So, what is meant by molecular phylogenetics? The study of evolutionary relationships among the biological entities, that is the individual population, species, or higher taxa, by using the combination of molecular data, like such as DNA sequences, then protein sequences, presence or absence of transposable elements, and the gene order data and these statistical techniques. So there are like molecular techniques and statistical techniques. Together we can call it as the phylogenetic analysis. It has all started from Fitch and Margoliouth in 1967. They made the first phylogenetic tree based on molecular data. If you see what is meant by phylogenetic tree, this tree was so close to already established phylogenetic trees that taxonomists realized the significance of molecular data, and this made them understand that other traditional methods that are although important, but molecular evidences could be final or confirmatory evidences, because the knowledge that is already stored in the DNA material, it is the foolproof or reliable data. Without any observational characteristic or the physiological characteristic, we can 100% we can rely on molecular data. The phylogenetic studies assess the historical processes which affect relationships, and phylogeographic studies that assesses the geographical distribution. The phylogenetic and phylogeographic studies started with the introduction of mitochondrial DNA markers in the population genetic analysis. So why mitochondrial DNA? DNA which is present in the mitochondria is most conserved in the animals, and so the animals which transferred in the ancient period of time from one location to other location, these organisms, they share almost maximum percentage of these DNA material, and they show similarity from different geographical area. The percentage similarity of these genes, at least we can understand based on mathematical values, and we construct phyto or phylogeographic structure or the data. The objectives: reconstruct the correct geological or genealogical ties among the biological entities, estimate the time of divergence between biological entity, and chronicles the sequence of events along evolutionary lineages. Now there are different molecular markers are there. Type 1 and type 2 are molecular markers. We have classified them. Type 1 markers are associated with the genes of known functions, and type 2 markers are associated with the genes of unknown functions. Then the terms comes of allozymes, microsatellites. Now what are they? Allozymes markers are the type 1 markers. We know them. Okay. These are as the proteins. They encode the associated with some sort of functions. Okay. And the microsatellites or the other neutral markers, type 2 markers, unless they are associated with the genes of some known functions, they are unknown. Now, allozymes electrophoresis is the method which can identify genetic variations at the level of enzymes. So different organism produces different types of enzymes. So based on these enzymes, we can identify or we can characterize the organism based on their enzymatic level and the types they contain that are directly encoded by these genes or the DNA material which is present in that organism. Protein variants called allozymes originates from allelic variations. They will differ slightly in electric charges, but there remains the same function with the variations in their structure. The functions will remain same, but the sequences will be different. As so, if the sequences will be different, ultimately they impose these changes on the structural level. And so if the changes in the structural levels of enzyme, we call them as allelic variations. Some alleles get changes. Now what is meant by allele? Allele is the gene which is having a particular characteristic, phenotypic characteristic. That's what we call it as allele. Allele could be a group of gene or a single gene. Okay, now remember that thing. Allozymes are codominant markers. Now, codominant markers having been expressed in the heterozygous individuals in the Mendelian way. Now, codominancy means what? Either they could produce, okay, either they transmit from parent to their progeny and sometimes they express, sometimes they go in the recessive state. And that state is, we call it as codominancy. The mitochondrial DNA markers, the other characteristic or the things that based on that we can understand the taxonomy. The mitochondrial DNA is a non-nuclear DNA because which is not present in the nucleus. It is present in the mitochondria. And the mitochondrial DNA is maternally inherited with the haploid genome. Now, what is meant by maternally inherited? See, guys, uh, when the reproduction happens, when sperm fuses with the ovum, what happens? Sperm only contributes genetic material. Okay, the head portion of the sperms only enters in the ovum of the female of that species, and so at that time, whatever mitochondria which is present in the sperm, it is present in the middle segment of the sperm and tail region. So no any mitochondria is actually enter into the ova. But if you see the ovum which contains already maternal mitochondria or the mother which contributes the mitochondria, ultimately these mitochondria will inherit all the children's. So whatever mitochondria we contents or that progeny contents, it ultimately belongs to maternal genes or maternal mitochondrial genes. The entire genome undergoes transcription as one single unit. They are not subjected to recombination and thus they are homologous markers. Now next one is microsatellites. The microsatellites is the simple DNA sequence which is repeated several times across various points in the DNA of an organism. These repeats are highly variable and these loci can be used as a marker. Single nucleotide polymorphism or SNP. Now SNPs arises due to single nucleotide substitution, either because of transition or transversion, or a single nucleotide insertion or deletion. This point mutations gives rise to different alleles with alternative bases at the particular nucleotide position. Single nucleotide polymorphisms are the most abundant polymorphisms in the genome, either coding or non-coding of any organism. These single nucleotide variants can be detected using PCR, microchip arrays, or fluorescence technology. With the help of these technology, we can understand SNP or single nucleotide polymorphism. DNA microarrays or DNA kits. Now, DNA microarray consists of small glass microscope slides. These slides, which is made up of a silicon chip or nylon membranes with the many immobilized DNA fragments arranged in a standard pattern. The DNA microarray can be utilized as the medium for matching the reporter probe of a known sequence against the DNA isolated from the target sample, which is of unknown origin. So species-specific DNA sequences could be incorporated to the DNA microarray, and this could be used for the identification purpose. The DNA extracted from the target sample should be labeled with a specific fluorescent molecule and hybridized to the microarray DNA. When the hybridization is positive, a fluorescent signal is detected with the appropriate fluorescence scanning or imaging equipments. Then arbitrary nuclear DNA markers. Now arbitrary markers are used when we target a segment of DNA of unknown function. The widely used methods of amplifying unknown regions are called it as random amplified polymorphic DNA and amplified fragment length polymorphism. Okay, so these two DNA markers RAPD and AFLP is used to identify the DNA of unknown function, and we can locate whether this DNA material or whether these characteristics are present within the organism or not. Specific nuclear DNA markers. Now these are called it as VNTR, that is variable number of tandem repeats. It is the segment of DNA that is repeated tens or even hundreds to thousands of times in the nuclear genome. They repeat in a tandem, vary in number in the different loci and differently in the individuals. There are two main classes of repetitive and highly polymorphic DNA. Mini satellite DNA is one of the other type of specific nuclear DNA marker. It refers to genetic loci with a repeat of 9 to 65 base pairs and microsatellite DNA with the repeats of 2 to 8 base pairs, which is very small and long. Microsatellites are much more numerous in the genome of vertebrates than mini satellites. Now next one is express sequence tag. Express sequence tags are the single pass sequences which were generated from random sequencing of cDNA clones. Now, what is cDNA? It's a complementary DNA which is being produced from the DNA material or the mRNA material through transcription procedure, either artificially or naturally. The express sequence tag can be used to identify genes and analyze their expression by means of expression analysis. The fast and reliable analysis can be made for the genes expressed in the particular tissue types under specific physiological condition or developmental stages. Different Different expressed genes could be identified using the cDNA microarray in the systematic way, and which is the very efficient techniques, and currently all the researchers or the institutes used to prefer this technique, the EST labeling or EST identification. These are the most valuable for linkage mapping. Advantages of molecular data. Now molecular entities are strictly heritable. The description of molecular characteristic is unambiguous. There is some regulatory to the evolution of molecular traits. Molecular data are amenable to quantitative treatment. Homology assessment is easier than with the morphological traits. Molecular data are robust to evolutionary distance, and it is also abundant. So it's less time consuming. That's why these are the advantages of molecular data we used to prefer for molecular taxonomy and analysis. The next one is DNA barcoding. Now, what is meant by DNA barcoding? It is the PCR amplification and sequencing of a particular genetic markers, usually the mitochondrial cytochrome oxidase 1 gene. COI means cytochrome oxidase 1 gene. Advantages widely used in arthropod identification. Whatever insects or the class arthropod belongs to this class, all the organisms we generally do barcoding and we can delineate them. Genetic or generic primer available for cytochrome oxidase 1 barcode region is generally useful for distinguishing closely related and less closely related taxa. Alternate markers can be sequenced if cytochrome oxidase 1 barcode is not differential or not preferable. Like take example of 15S rRNA or 12S rRNA. Likewise, we use 16S rRNA in microorganism. We generally use 12S rRNA and 18S rRNA in arthropod species. There are ample disadvantages are also there. Like take example, if we count them or if we list them, the first comes up like it requires large database of sequences for comparison. The prior knowledge of barcoding region is required when applied diagnostically. Without any knowledge of cytochrome oxidase 1, we cannot make the primers of these genes, COI genes. So how could we do PCR? So we should know at least the initial and final segments of the cytochrome oxidase genes or cytochrome oxidase 1 gene, so we can make primers and we do PCR systematically. Individual sequences may not provide the sufficient discrimination when studying the cryptic species complexes. Cytochrome oxidase 1 and other mitochondrial genes are maternally inherited particularly, so which may result in decreased barcoding diversity because only one lineage from female to female or the female segments of that species only we are tracking down. So the skew phylogenetic inferences is there. So what is meant by specific PCR? Targeted assay giving a presence or absence of result of particular genus or species. Take example of RT-PCR that we generally do in the corona. We specifically segment the two types of genes from these coronaviruses and we amplify them and we see whether the virus is present or not. So it is completely a typical specific organism based PCR. So PCR is always specific. Okay. So targeted assay gives us presence and absence of result. There are advantages are there. Useful diagnostically if as it targets the specific taxon, can be used to target of specific genus, species or strain within a mixed sample. No sequencing of PCR product is required, only with the PCR. So we are not sequencing the whole coronavirus, right, to identify whether it is corona or not, but we only target few sets of genes which is specific in the only coronavirus, that is SARS-CoV-2, or currently now what we are calling them as mutated Omicron and Delta or Omega viruses, right? So we particularly understand the specific sequences or the mutated sequences. Accordingly, we design a primer, and with the help of this primer, we can apply to real-time PCR or reverse PCR. With this PCR technique, we can identify if there is an amplification of that particular segment. Of course, the organism is present. If there is no any amplification, the organism is absent. Disadvantages: require specific primer design, assay optimization is required, then specifically test and priority to use to diagnostic tools is very much important. Then another is size differential PCR. Now what is meant by size differential PCR? It employs generic PCR primers but yields amplicons that differ in length, usually target the intergenic transcribed spacer region, that is called it as ITS. There are advantages like discriminate between range of species simultaneously. Differentiation, it's by electrophoresis mechanism also we can differentiate them or we can identify them. Does not require sequencing or the amplicon. Advantages, disadvantages are there. Size of amplicon needs to vary substantially to the enamel discrimination. ITS regions contain repetitive regions that can result in PCR products with the multiple bands. So these multiple bands can be problematic to understand or to identify whether that species is or whether that species is there or not. The next one is Pcr Rflp. We can Pcr the restriction fragment length polymorphism or these markers with the help of specific marker DNA material. They are the genetic marker, so they are size variable markers. Okay, and the level of discrimination is very good actually. So size does not matter at all. It depends on again the PCR technique and reverse Taq polymerase enzyme, whether the which kind of Taq polymerase we generally use. Generally, there are Thermus aquaticus organism, the enzyme that is what we have isolated from Thermus aquaticus by Carey Mullis. We generally use for smaller segment. For larger segments, we use the another organism, that is preferengens. If you know, the that is also called it as PUF polymerase enzyme, PUF. I hope so. You just go through the several reference books. You will understand what different polymerases enzymes are there. There are certain types of disadvantages like the downstream digestion, amplified DNA mutations may occasionally result in unidentified RFLP pattern. Multiplex PCR is there. They combine multiple primer sets with the different specificities in the single assay. There are certain advantages and disadvantages. RAPD is there. RAPD is very important because it is a randomly amplified polymorphism. Okay, polymorphic DNA material. It uses random primers. So use just random primers and identify the RAPD material within them. And based on RAPD fragment production or the amplification, we can electrophorese them and we can check the patterns of RAPD within the electrophoresis. And so within that we can understand the RAPD patterns of that organism. There are certain advantages and disadvantages also there. FLP is there. Microsatellite analysis is also there. So development of microsatellite. How do we do microsatellite? We extract the genomic DNA of different species. We digest the DNA with the help of restriction endonucleases, and after the digestion of these restriction endonucleases we can get a specific fragments of DNA material. We ligate them into the particular vector molecule or plasmid vector molecule. We transform these bacteria into the plasmids into the bacteria, the host bacteria. Like take example alpha E. coli or these I forget now the name of that bacteria, but it's the E. coli strain which we generally use to for as a host organism. Uh, the competency of this bacteria is very good. So once the engulfing of plasmid is there, we transform them. Transformation takes place. We perform the hybridization experiment with the microsatellite specific probes, and we sequence the positive clone, and we can get what different microsatellite portions or are there or not. We label these probes with the radio labeling or fluorescent labeling, and then we can understand, or with the help of southern blotting we can understand the sequences. Then quantitative PCR, like I said, RT-PCR is the quantitative PCR where cyber green dye is used. That is also called as double dye. LAMP is one of the technique. Now see guys, LAMP is new technique. Loop mediated isothermal amplification. Now what is it? Just pay attention. We generally employ three sets of specific primer. We use these specific primers for amplification under such a isothermal conditions, and within the isothermal condition whatever amplification we yield, we actually use ladder, yields a ladder of amplicon on the electrophoresis, or the amplicons can be detected by cyber green dye, and we identify what different fragmentations are there or what different sequences has been amplified. So this is called it as, remember, loop mediated isothermal amplification, LAMP. So many in the so many competitive examination in gate or net or pet, generally it comes, the question comes up like, uh, what is the type of this technology, the lamp, what it is, what is the lamp, and the option will be like that, it is the PCR, it is the diagnostic methodology, it is the X-ray diffraction methodology, what it is. Remember, it's the PCR methodology. I hope so you understood the what different tools and techniques we generally use in taxonomy and characterization. Thank you very much.